Welcome back to Chapter 8, Nucleotides and Nucleic Acids. In this lecture, we will be discussing about nucleic acid chemistry. This involves DNA denaturation, non-enzymatic transformation, and DNA sequencing. Just like proteins, superstructures of nucleic acids, such as DNA and RNA, have physiochemical properties. DNA and RNA are highly negatively charged. DNA double helices, as well as secondary structures, can fold and denature, just like proteins. In case of DNA, this folding and denaturation is reversible. What does that mean? The covalent bonds remain intact in DNA when they undergo denaturation. The genetic code remains intact. That's what it means. As you can see in the image, there is a double helical DNA. And upon denaturation, as the arrow goes down, you can see that the individual strands begin to separate. And that's what happens in partially denatured DNA. As it continues to denature, the strands separate from one another and they become random coils and are no longer double helices. How does this happen? Because the hydrogen bonds are broken and hence two strands separate. The base stacking is lost, and so UV absorbance increases for individual random coil single strands. Couple more points. Denaturation can be induced by high temperature or change in pH. And this denaturation, going from a double helical DNA to separated single-stranded random coils, this can be reversible. And the reversible process is called annealing. As you can see, as you go up by looking at the arrow, association of these two strands by base pairing can lead to partially denatured DNA, which then undergoes annealing to form a double helical DNA. So these are some of the important points of thermal DNA denaturation. DNA exists as double helix at normal temperatures. For example, room temperature at 25 degrees centigrade. Two DNA strands dissociate at elevated temperatures. If you increase the temperature from 25 degrees Celsius to, let's say, 70 or 90 degrees Celsius, the double helix begins to separate or denature. Two strands re-anneal when the temperature is lowered. So you increase the temperature, you can denature the DNA. You decrease the temperature, they'll start annealing. The reversible thermal denaturation and annealing forms the basis for what is called as polymerase chain reaction, or PCR. DNA denaturation is commonly monitored by UV spectrophotometry at 260 nanometers. The reason why 260 nanometers is because of the absorbance of purines and pyrimidines. In this slide, we will discuss some factors that affect DNA denaturation. The midpoint of transition, or TM, as shown in this figure, is the melting point, and it depends on base composition. As you can see here, the plot of denaturation versus temperature for two different DNA specimens. For DNA specimen A, which gives a blue curve, TM gives the transition melting point. And for a different specimen, which is given by a red colored curve, TM is the transition or melting point for that DNA specimen. Now, this depends on base composition. As I said, high GC or CG composition increases TM, which means that if you have among these two bases, The base with the red colored curve seems to have a higher GC content. And if you see at a plot of G plus C, this percentage of total nucleotides containing G plus C versus temperature, as you can see, Tm increases linearly with increase in GC content. Tm also depends on DNA length. Longer DNA has longer Tm. It's important for short DNA. Tm also depends on pH and ionic strength. High salt content increases Tm. Two near complementary DNA strands can hybridize. For example, let's consider an experiment with two samples, sample 1 and sample 2, that have near complementary sequences. If you were to mix these two samples and cool them, you can actually obtain hybrid duplex DNA of sample 1 and sample 2, in addition to duplex of sample 1 and duplex of sample 2. This is a very important experiment because this has various different advantages and applications. What are they? This can be used for detection of specific DNA molecules in complex mixtures, such as radioactive detection, fluorescent DNA chips. All of these utilize this technique of DNA hybridization. One can amplify specific DNA sequences of whichever one wants or needs. It can be done using polymerase chain reaction. Remember, polymerase chain reaction involves heating and cooling DNA strands. Involved in site-directed mutagenesis. We'll talk about what site-directed mutagenesis in a little bit. Let's discuss about mutations. A mutation is a change in the DNA sequence. This will sometimes lead to a DNA, RNA, or a protein having a defect. Mutations can happen in two ways. One, DNA that is chemically modified is a mutation. Errors in transcription or replication can also lead to mutations. Now, error in transcription can lead to mutations in RNA, which can further lead to mutations in proteins. Here are some of the molecular mechanisms of spontaneous mutagenesis. First, deamination. It's a chemical transformation, and deamination is when an amine from a base, it could be either purine or pyrimidine. This is just an example. This amine gets converted to a carbonyl. So this is a deamination where this amine is replaced by an oxygen. These are very slow reactions, but you have many residues in DNA. So mutation of one specific base is good enough to have a mutation in the translating protein. The net effect is significant because of this. There are about 100 cytosine to uracil events that happens every day in a mammalian cell. That's a lot. The next event is depurination. The N-glycosidic bond is hydrolyzed. For example, in case of guanosine, as you can see, this bond is the N-glycosidic bond, where this nitrogen is connected to the ribose, can get hydrolyzed to give guanine and an apurinic residue. This is significant for purines. About 10,000 purines are lost per day in a mammalian cell. Cells have mechanisms to correct most of these modifications. It's a good thing. Next, let us consider molecular mechanisms of oxidative and chemical mutagenesis. So for oxidative damage, hydroxylation of guanine is an example. And this happens in mitochondria, and mitochondrial DNA is the most susceptible. For chemical alkylation, guanine gets methylated. Now, even though these modifications do happen in our body, cells have mechanisms to correct most of these modifications. However, cells can miss many different mutations that happen in our body. That is a big concern. Let us consider molecular mechanisms of radiation-induced mutagenesis. UV light, predominantly from sun, induces dimerization of pyrimidines. This may be the main mechanism for skin cancers. Ionizing radiations like X-rays and gamma rays causes ring opening and strand breaking. These are relatively difficult to fix. Like the other two mutagenesis, cells can repair some of these modifications that are caused by radiations, but others cause mutations. Accumulations of mutations is linked to aging and carcinogenesis. This figure explains the formation of pyrimidine dimers induced by UV light. And this is one type of reaction that is shown here that results in the formation of a cyclobutyl ring, as you can see. In the normal arrangements of pyrimidines here, this is two thymines, it can result in this cyclobutane ring formation among these two stacked thymidines. An alternative reaction results in a 6,4 photoproduct with a linkage between C6 and a C4 of the adjacent thymine. Formation of a cyclobutane pyrimidine dimer like this introduces a kink or a bend into the DNA, the double-stranded DNA, and this is deleterious. In contrast to mutated DNA, there are some modified DNA that is physiologically relevant. DNA bases can be modified for functional reasons. Bacteria, for example, uses methylated DNA to distinguish itself from invader. Now, the invaders can be DNA that can get into bacteria, as well as in handling of mismatched DNA. Eukaryotes, on the other hand, use methylcytosine to prevent transposon migration. Next, let's discuss one of the most important application of DNA analysis, and that is DNA sequencing. Frederick Sanger developed a sequencing method using selective digestion of DNA dideoxynucleotides. They're called as DDNTPs, and I'll tell you in the next slide as to what they are. DNA synthesis are enzymes that make new copies of DNA, and for sequencing purposes, DNA synthesis can be used to make new copies of DNA that you are interested in sequencing. These incorporate a small fraction of DDNTPs at each position terminating the strand. I'll explain to you in detail in the next slide. Then the base-specific cleavage are performed and sequence is determined by gel mobility. This is what is called as the famous dideoxy sequencing of DNA. This figure shown here explains as to how a DNA synthase works and how this can be applied to sequencing DNA. Now, there are two strands here. One is a template strand and another one is a primer strand. The template strand is the strand that one wants to sequence. A primer strand is a complementary region of a part of a template strand, which is shorter than the template strand actually. If you can look at the sequence GATTCG, it's complementary to CTAAGC. Now what a DNA synthase does is it adds nucleotides to the remaining of this template strand by using the primer strand as a guide. So it can bind to this primer strand as well as the template strand, and it sees that there is a pattern, and based on that it can add complementary bases to these. For example, in the place of the complementary base against T, G against C, C against G, and T against A, G against C, and A against T. So this principle or this feature of DNA synthesis was recognized by Sanger, and he utilized this to develop a sequencing method. This ingenious method makes use of a nucleotide called as DD-NTP, or NTP means nucleotide triphosphate. In this case, it is DD-ATP. And what DD means, dideoxy, which means both the hydroxy in the ribose is removed. Remember, in DNA, only the 3' hydroxy is present. The 2' is not. But DD-NTPs will have both hydrogens. Now, the cool part about this technology is that when DNA synthase works on the template strand in making it using a primer strand, and if you give a DD-NTP, for example DD-ATP, it uses it and attaches against this thymine. But the moment it does that, it cannot attach the next sugar because a 3' hydroxy is required for it to attach the next sugar, as shown here, right? That is the hydroxy that attacks the phosphate and makes a covalent bond like this. So in a DD-ATP, that hydroxy is absent. So DD-ATP terminates synthesis because the 3' hydrogen cannot act as nucleophile in phosphodiester bond formation. And so how can this be utilized to sequence? So what Sanger did is that he used various different DD-NTPs like DATP, DCTP, DGTP, and DTTP in this template versus primer. For example, template would be a DNA that is unknown and that needs to be sequenced. And there would be a generic primer that he would use, and he would then map the rest of these nucleotides. The way he does is when you use this mixture of these nucleotides, for example, I'll take you through two of these. For example, if you use DATP, and if you imagine that this is the sequence of a template from 3' to 5', what a DNA synthase would add opposite to CTAAGCT would be GATTCGAGCT. This is just the complement. This is just a complement of this, right? Now, if it uses a dideoxy ATP in place of a nucleotide, it could, in principle, add a DD at the last adenine, or so it could be this adenine, or this adenine, or this adenine. There are 3 adenine. So the 3 possibilities: it could add to the final one, or it could add to the central one, or the first one. It cannot add all 3 of them, remember, because if you add one DD-ATP, the next base is not going to form or attach. The same way with DCTP. Wherever you see a C, right? Those are the places where DDCTP can be added. So this is the ingenious uh, tactic or the strategy of this method is that you know exactly what you're getting into. Then, once you have these various different small DNA fragments formed by a DNA synthase, you collect them and run on an agarose gel. And when you run on an agarose gel, and one can actually look at this gel because it can you can actually follow the radioactivity of the autoradiogram, or the radioactivity of this specific label. And I forgot to tell you that there's a label that is incorporated, and that is a tritium. That tritium molecule is radioactive. Because of that, one can follow using an autoradiogram. Now the sequence of the complementary strand is shown here, and the way the sequence the way this DNA is sequenced is from the bottom. This is G because it's ACGT as shown here in the vertical scale, and the bottom, as you can see from the bottom, this is G A T T C G A G C T G A. That's exactly the sequence. And from the gel, one can deduce the sequence of the unknown DNA. This is exactly how Sanger sequencing works. Sanger sequencing is currently automated. So in the Sanger method, each ddNTP, or 4 of them, can be linked to a fluorescent dye, as shown here. The fluorescent dye gives the same color to all fragments terminating in that nucleotide, with a different color for each nucleotide, as shown here. The sequence is pretty similar in the sense that a DNA synthase is used for a template of unknown sequence, and the primers are attached with the fluorophore and a ddNTP of different color. All 4 labeled ddNTP are added to the reaction mixture here, and they're mixed together. The resulting colored DNA fragments, as you can see here, are separated by size in an electrophoretic gel, as you can see. And this electrophoretic gel is in a capillary. So it's a capillary gel in a single band. And the color associated with each band, as shown here, is detected with a laser. So this capillary is inserted in the path of a laser beam, which then is detected by a detector. Now, the DNA sequence is read by identifying the color sequences in the bands as they pass the detector and feeding this information directly to a computer. The amount of fluorescence in each band is represented as a peak in the computer output. That's what you would see. And based on the color, each peak is given an identity, and each peak corresponds to individual nucleotides. And depending on the migration of these different fragments, the computer then decodes it for a sequence. And that's what you see here. This is the modern sequencing technique. And this is adapted from what Frederick Sanger had developed originally. In addition to their roles as the subunits of nucleic acids, nucleotides have a variety of other functions in every cell. For example, they function as energy carriers, components of enzyme cofactors, and chemical messengers. To start with, cellular energy is often stored as adenosine triphosphate, or ATP. And shown here is a structure of ATP. And remember, it has a ribose sugar ring with an adenine base. There are also GTP CTP, and UTP. ATP is for the most widely used nucleotide triphosphate, but other nucleotide triphosphate are also used in many reactions. Nucleotide triphosphates also serves as the activated precursors of DNA and RNA synthesis. Now, if you look at this structure of this nucleotide triphosphate, it has an anhydride bond and an ester bond. Now, the typical structure of acetic anhydride is this, whereas an ester is this. Now, anhydrides are high energy bonds, and hydrolysis of an anhydride bond yields more energy than hydrolysis of ester. A carboxylic acid anhydride, for example, cleavage of this gives more energy than cleavage of an ester. Similarly, cleavage of an anhydride phosphorus anhydride bond gives more energy. So the energy released by the hydrolysis of ATP and other nucleoside triphosphates is accounted for by the structure of the triphosphate group. In addition, hydrolysis of ATP to ADP or ATP to AMP releases enormous amount of energy. This is why ATP is called as a high energy molecule, because ATP hydrolysis to ADP leads to a release of about 30 kilojoules per moles of energy. Hydrolysis of ATP to AMP, on the other hand, leads to a release of about 45 kilojoules per moles of energy. Nucleotides also function as cofactors of enzymes. Modified adenosine function as enzymatic cofactors. The three most common cofactors are coenzyme A, nicotinamide adenine dinucleotide, and flavin adenine dinucleotide. Coenzyme A, the structure of which is shown here, is an important cofactor. The adenosine portion is shaded in light red, as you can see. CoA, as it is called in short, functions in acyl group transfer reaction. The acyl group, such as acetyl or acetoacetyl group, is attached to the CoA through a thioester linkage to the beta-mercaptoethylamine moiety. So usually the acyl or the acetyl group is attached to this sulfur. Coenzyme A is an important cofactor in fatty acid and lipid biosynthetic pathways. The other two cofactors that involve adenosine are NAD+ and FAD. NAD+, as you may have heard about, or you may know, functions in hydride transfer reactions, and the nicotinamide ring can undergo reduction, and this is the business end of this cofactor. Similarly, FAD or flavin adenine dinucleotide is an active form of vitamin B2 or riboflavin, and it is involved in electron transfer, and this is the business end of this cofactor. Nucleotides also act as signal molecules. Many cyclic nucleotides act as signals. They are typically part of a signaling cascade that begins with binding of a ligand outside the cell. This induces a change in the transmembrane receptor, which stimulates production of what is called as CAMP or cyclic adenosine monophosphate. As you can see in the slide, CAMP or cyclic adenosine monophosphate is formed by an enzyme called as adenylyl cyclase. And adenylyl cyclase is activated by a signal that comes from outside the cell, which converts ATP to CAMP. CAMP is a messenger molecule or a signaling molecule that initiates a cascade of reactions. We'll talk about biosignaling pathway in advanced biochemistry course.